Good afternoon. My name is Alison Menzies, and my presentation today is about a study I undertook on the biology and larval pupal parasitization of the painted cup moth Dorytifera oxlei in the family Limicodidae. This study was completed as part of a third-year research project during first semester this year at La Trobe University with Martin Steinbauer as my supervisor. I'd like to begin by acknowledging the traditional owners of the land on which we meet today, respectively, and those on whose land this study was conducted, the Dja Wurrung. I would also like to pay my respects to elders past and present. And also on this slide, shout out to the special appearance by the Perthida moths in the background of my intro photo. There are 8 Australian endemic species of the genus Dorytifera. As with most Limicodidae, they are noted for being slug-like in appearance, and Dorytifera in particular are noted for their aposematic colouring, urticating setae, and their cup-shaped cocoons. Now, we all tend to recognise these larvae, but there are very few studies of the genus. They primarily appear in literature in relation to their capacity for outbreak defoliation, but Australian Limicodidae studies number around 7 or 8 in total. I think there's a new one about a month ago. So as an outbreak defoliator, this genus typically defoliates 50 to 90% of canopy. They do tend to favour eucalypts, but Dorytifera stenora usually herbivorizes mangroves. The more notable documented defoliation events, however, are by painted cup moths. In 1976, in East Gippsland, it defoliated around 25,500 hectares of native forest. And in 2019 Hobart, they also defoliated thousands of hectares of local eucalypts. These 2 images here are of the same section of yellow gum in my driveway. The top is November last year when painted cup moths nearly stripped the entire tree. And the bottom is early March this year, where it has had somewhat of a chance to recover. So as you can see, there's cause for concern in eucalypt forests from this genus. In this study, we focused specifically on Dorytifera oxlei, the painted cup moth. This was intended to be a natural history type study, hopefully as a prelude to a more rigorous population ecology study at some point in the future. Again, although the larvae of this species is highly recognizable, most data on it is anecdotal. But what we do know is that it is likely univoltine. Eggs are laid March to early May and larvae hatch April, May. They feed through winter until late spring. Pupation takes place over roughly 2 weeks in mid to late November, and the adult moths close in March to late April. Early larvae are gregarious and they likely go through 7 or more instars. And as late larval instars, they begin to differentiate and become sexually dimorphic. In these images here, the larvae on the left is a male cup moth and on the right is a female. The females end up about 1/3 as large again as the males. The adults too are sexually dimorphic. They're also, this here is a male. They're also likely semiparous. While not all eggs are laid in the one clutch, up to about 4 or 5 clutches tend to be laid close to each other by the female and then she dies. In this study, we aim to investigate the top-down population regulation of cup moths with a focus on potential parasitization. The study was conducted in Castlemaine in the Goldfields region of central Victoria where I live. We also hope to see if host tree played any part in the types of or presences of endoparasitoids. Given that where I live, this species is displaying characteristic traits of an outbreak lepidopteran, we wanted to see if there were accompanying and appropriate top-down biological population controls. Further, given that this species has absolutely no compunction about stripping the yellow gums and ironbarks at my house, this study was somewhat personal. Again, most of the accounts of population controls of painted cup moths are anecdotal. There are accounts of diseases that turn them to sludge that are present in 1 or 2 sort of general gardening books. Predators are likely in the form of a couple of opportunist native bird species and some Hemiptera. Endoparasitoids have been more reliably documented in studies of Hymenopteran and Dipteran families. So in early December 2020, I collected around 183 cocoons from on, under, and around and inside bark curls from 4 eucalypt species in Castlemaine. The aim was for 200, but some of the collected cocoons, I think, turned out to be the previous years. The host trees were a Eucalyptus cameldalensis, a river red gum, an Eberania, which is a bull mallee or broadleaf box, and what turned out to be 2 Elocoxylons, 2 yellow gums. The first 3 trees were from my house. The second yellow gum was on a friend's property nearby. These 2 yellow gums had very different habits, and I also didn't actually ID the trees until after the cocoons were collected, which is kind of why we ended up with 2 yellow gums. So each cocoon was weighed. They were kept in individual containers. They were lightly misted every 3 days and monitored daily for emergencies. So when we weighed the cocoons, we found that the weights reflected the sexual dimorphism of the larvae. As you can see here, there's a pretty clear differentiation between the female and the male moths. With cocoon weight as a dependent value, host tree was found to be an influence. And when host tree and gender were also taken into account, there was a difference in weights based on an interaction of host tree and gender. And this was an interesting result at this early point of the study. So once all emergencies and eclosures seemed to be over, we conducted a census on all cocoons on the 5th of May, 2021, this year, in which we opened all unenclosed cocoons and determined outcomes for them all. So 137 of the cocoons contained adult moths, so 76%. 9 of the cocoons had prepupate larvae, so they were still in larval form. 13 had been parasitized by wasps, which is just over 7%, which was a nice outcome. And another 22 were what we called unknown, as the contents were kind of desiccated or a nice black smelly sludge. It could be that these were from the year before or that they had been affected by disease. So when we looked at moth gender, we found that out of 137 adults, there were 70 males and 67 females, which shows no real departure from usual gender splits in Lepidopterans. The male to female splits, as you can see, with most of the trees is roughly 50-50. And we found that there was no host-related effect, and this was a fairly straightforward result. When we looked at the wasps, we found that they were all the same species, which was an endoparasitoid Eurytoma, which we could only identify down to genus. These wasps are a recognized endoparasitoid of a few orders and have been specifically recorded as an endoparasitoid of the painted cup moth. So the endoparasitization rate was 7%, with a total of 407 individual wasps recorded. What's sort of important to note at this point in the study is that the emergence dates for these wasps was from the 3rd to the 28th of January this year. And as I sort of previously mentioned, the adult moths don't actually emerge until March. So when we look at parasitization, the gender for the wasps was around 1 to 2 male to female, so sort of standard-ish for Hymenoptera and Eurytomids. We did have different rates of parasitization on the 4 host trees. So that sort of ranged from 4 to 16%. But what was most interesting was that there was no parasitization at all on the first, or on one of the Elocoxylons. So this was a tree that actually had the most cocoons collected from it. And the parasitization rate for the other Elocoxylon, the other yellow gum, was actually the highest for the lowest number of cocoons collected. So this result was unexpected and provoked much speculation. So from all this data, we are now able to build up the following overview of the life history of painted cup moths in the Goldfields region. So in late November to December, the larvae began pupation. We saw endoparasitoids emerge throughout January. So roughly 4 weeks later, in late February, the cup moth adults began to eclose, and they continued to eclose and lay eggs until late April. Larvae feed from autumn throughout winter and spring until pupation. We suspect that the Eurotomids parasitize sometime in spring, although they do have quite a wide window for it. So the main conclusions that we can draw from this study are that host tree has some effect on cocoon size and parasitization rates. Sex ratios were confirmed as standard for both species families. The Eurytoma wasps are a top-down population control of cup moths, but the relationship dynamics are quite uncertain. They're unlikely to be exclusive specialists on painted cup moths, as there's at least a good 8 weeks or more likely 7 months before larvae are at a suitable stage of development for Eurytoma to parasitize. So future questions to look at in the short term, is the Eurytoma wasp endoparasitization opportunistic or a regular occurrence? Eurytoma are probably generalists, which is why they're not good regulators of painted cup moths with a rate of 7%. And superficially, the data around host trees supports that these wasps are generalists. And in what way do host trees specifically affect parasitization in terms of age, species, and provenance? And long-term questions, given that cup moths seem to be an outbreak species, we should be looking at how long these outbreaks last for and how cyclical they are as well. One study I looked at estimated that there was 15 years between cup moth outbreaks, but the data that was used was originally for a leaf longevity study, so more specific data on that would be useful. And so with that, I hope you enjoyed this modest history study. Thank you very much to my supervisor, Martin Steinbauer, and to Andy Austin and Erin Fagan-Jeffries for all their WASP ID assistance. I'm happy to answer questions, or if there's no time, please feel free to email me, and thank you very much for your time.
Well done, Alison, for your first ever presentation at a scientific conference or an entomological scientific conference. Very good. We have 3 minutes for questions, so you have plenty of time for to see. We'll see what sort of questions we get. And Stacey has joined us too. So, in the absence of any other questions at the moment, what might you think is the reason a high number of larvae and pupae escape parasitization on that host?
Well, as you've warned me, I need to be very generalist about this. It's likely, as I mentioned, that they're not exclusive parasites on the Dorotifera oxlei. One of the reasons I think that why one of the leucoxylon trees might have escaped parasitization is that was not necessarily an endemic species. It was planted in our driveway, so it may be a dwarf. I think it's called yellow box. Maybe they may not know how to find. They may not know to find that tree. I'm really not that sure. More study is definitely needed.
What would you do if you're going to rear this thing again? What if people could now start, but it seems we possibly were keeping them too dry? Is that possible?
It was possible that I didn't water them enough as they possibly needed. Perhaps possibly cheesecloth instead of keeping them in sealed containers would be the best method, I think, of keeping them alive for 6 months. Or 3 months.
It's really important stuff. All this basic biology of all these outbreaking species about which we know almost nothing. And this is the 21st century. It just never ceases to amaze me. So we still can't explain fluctuation in populations of any of the major defoliators of Australian forests. Quite impressive for the 21st century, I reckon. But anyway, I'll stop carrying on.